Hello, everyone. My name is Susan Seegers, and thank you for joining this session on AOCS official methods. This session focuses on procedural and technological updates to current AOCS methods that reflect the changing scientific landscape. Updates from the technical committee will also be discussed. We've got two great presentations for everyone today. First, we have an introduction on new AOCS methods and methods under development from Scott Bloomer. Then we'll also have a presentation on the statistical treatment of proficiency testing data, how to interpret laboratory proficiency reports. So first, with the new AOCS methods and methods under development, we have Dr. Scott Bloomer from AOCS. Scott Bloomer started his research career in 1973 in the laboratories of Honeywell, Incorporated, doing research in humidity sensors. Since then, he's been privileged to do research on smoke detectors, biosensors, enzymatic interesterification, basic oilseed processing, and the application of enzymes to milk compounds. He's written a dozen peer-reviewed papers and is a contributing editor for the AOCS trade magazine, Inform. Bloomer managed the fats and oils portfolio of ADM for several years, drafting and defending patents. He is currently the Director of Technical Services at the American Oil Chemists Society. Thank you.
Good morning. Welcome to the 2021 AOCS annual meeting. My name is Scott Bloomer. I'm the Director of Technical Services at AOCS. Today I'll be bringing you an update on AOCS official method development in the current year. First, let's talk about method types that AOCS has. There are 4. The most common of them is the AOCS official method, of which we have over 400 of them. A method proposer will bring their draft of a method to AOCS, and we will work with them to get it into the proper shape and a certain level of completeness, and then it's sent to a subcommittee of experts for review. These experts will ask several questions. Is the method clear? Is it complete? Can it be carried out without needing to consult with the person that wrote the method? And is it ready for a collaborative trial? When these questions are all answered yes, then the method goes on to a collaborative study. The person proposing the method often becomes the study director. Samples are selected and sent out, and the labs that are recruited will carry out the analysis. Following that, the data is sent back to AOCS, and AOCS carries out statistical analysis, throwing out outliers, and making sure that the study was carried out properly. When that's finished, everything is bundled together and sent back to the expert subcommittee for their final review and approval. Once that is obtained, the entire package is sent on to the Uniform Methods Committee of AOCS, which is our highest methods committee. And these experts put the final seal of approval on the method, and it can go into publication by AOCS. There are 3 more AOCS method types. The standard procedure undergoes the same level of rigorous review that an official method goes through. However, it uses only one instrument from one manufacturer, so it is a bit more limited. We also have recommended practices. This would be something that doesn't have as much of a broad review in the collaborative trial, or may perhaps lack a collaborative trial altogether. However, these methods are considered important and so can be published as a recommended practice. Finally, we have analytical guidelines. This would be a list, usually, of methods that are applicable to a matrix. So, for example, if somebody starts to produce something that they've never produced before, like lecithin, and they want to know which tests they should use, they can rely on the analytical guidelines. Since our last meeting, we've published several methods. Here we see a joint Japan Oil Chemists Society AOCS official method CD29D-19. This is for determining 2,3-MCPD fatty acid esters and glycidyl esters in edible oils using an enzymatic hydrolysis method. This was issued in October 2020. In addition, there's a recommended practice from the same lab, joint JOCS AOCS recommended practice CD29E-19, which is 2,3-MCPD fatty acid esters and glycidyl fatty acid esters in fish oils by enzymatic hydrolysis. This can be a very important question for marine oils, and so I'm looking forward to an expansion of this through a collaborative trial when things become ready. In addition, we have published a new official method, BA12A2020. It was built upon a previous method that was limited in scope to soybeans. This is a trypsin inhibitor activity method, and it was carried out by Dr. Keshen Liu under very interesting and trying circumstances, but he did an outstanding job, and he expanded the scope of the method to include pulses and cereals and other grains. So we're looking forward to seeing a broad institution of this method in our LPP. As usual, we had several methods that were revised in the last year. Two of them, spectrophotometric and ATR methods for transisomers, were recently revised. In addition, the trace elements, especially metals and oils by ICP, was exhaustively upgraded and expanded recently. And we also just revised the method which gives guidance on writing and approval AOCS methods. In addition, we also recently made a revision to a method for capillary GC determination of a broad spectrum of fatty acids, including polyunsaturated fatty acids. Let's talk about ongoing method revisions. AOCS method CE6-86, HPLC analysis of phenolic antioxidants, is undergoing a revision. Dr. Mark Collison from ADM is carrying out a collaborative trial right now to improve on this method. The old method had been used to make statements that a certain fat or oil didn't have any phenolic antioxidants in it, but it wasn't quite that good. Dr. Collison is improving the method in order to be able to make much better statements about the lowest levels of phenolic antioxidants, as well as steps to remove interference that can occur in canola oil. In other ongoing method projects, Professor Amir Taha has continued to work on his one-step method for extracting fatty acids. This will replace Fulsch washing and reduce the amount of chloroform needed quite a bit. He's in the process of carrying out a collaborative trial. In addition, Wilmar brought us a method for determining monochloropropandiols and glycidol in glycerol. This is unique. We don't have this method anywhere. And this uses GCMS, and the project is just about ready to go to collaborative trial. We have two oxidation methods that are being developed right now. The first, the RSSOT, which is a small-scale oxidation test, is very quick, and it's been brought to us by Anton Parr, and it's for determining the oxidative stability of fats and oils. The manuscript is almost finished. In addition, there's another accelerated oxidation test for determining the oxidative stability of feeds, foods, fats, and oils. This is brought to us by Microlab. It's called an OxyPress. This latter method purports to give an ability to calculate the shelf life of a food. That draft has been finished and is under review by one of our expert subcommittees. Here's an ongoing method project that we are working with in collaboration with the International Organization for Standardization, ISO. It's a high throughput method for 2,3-MCPD fatty acid esters and glycidyl fatty acid esters in fats and oils using GCMS-MS. It's actually a clever combination of a couple of earlier methods that works in a high throughput and a more rapid way. This has been given AOCS method number CD29F-21, and it's the same as ISO 18363-4. The two methods are going together in lockstep. The AOCS chromatography subcommittee first approved the method in 2019, and it's gone through ISO, and a collaborative study has been carried out. So in ISO, the draft international standard has been reviewed, and in AOCS, we are looking at it again, the post-collaborative study expert subcommittee review. Another project that we're working on with ISO is a collaborative trial for the fatty acid composition at the SN2 position of triacylglycerols mentioned earlier. This is joint JOCS, AOCS official method CH3A-19. We are in the process of planning the collaborative trial, and the ISO group SC11, which is for fats and oils, is working together with the ISO group SC5, which is milk products, and we have formed a joint working group and are working together for the first time in many years. We've had an initial review, and the experts are commenting right now, and if you're interested in joining the collaborative trial, which will happen later this year, please email me at this email address. Here's a new method project, and this is being brought to us again from Archer-Daniels Midland. This is a replacement of recommended practices CE 3-74 and CE 7-87 with a single method for measuring tocopherols and sterols in deodorizer distillates and similar matrices by gas chromatography. It has the benefit of very simple, minimal sample preparation and a 30-minute runtime, and there's a broad variety of materials that will be found in deodorizer distillates that this method will quantify. In addition, we are working with Dr. Miyazaki, who brought us the two methods that were mentioned earlier. Miyazaki has been working with the Japan Ministry of Agriculture, Forestry, and Fishes, and she has developed a method for GE and MCPD esters in finished foods, which is a method that's very much needed. This particular method uses pressurized liquid extraction, the EFSA method, which has been well established, and then after extraction, the lipids are analyzed by the joint JOCS-AOCS official method 29D-19 that Dr. Miyazaki brought to us. A manuscript has been prepared in English, and it's under review by the Ministry of Agriculture, Forestry, and Fishes, and once that's finished and approved, it will be sent to AOCS so we can start the process of putting it into the official method format. Finally, here's a new method project, determination of available lysine with liquid chromatography. This is a method for determining lysine, which is of course a very important and often limiting nutrient, in corn gluten meal, distillers dried grains of solubles, known as DDGs, soybeans, and processed products thereof. And the draft method is in preparation, and it is almost ready to be sent to the expert subcommittee for review. And this has been brought to us by Dr. Sorda Ime Latika. If you're interested in participating in the collaborative trial for this, please feel free to contact me. Thank you for your time. Can I answer any questions for you?
Thank you, Dr. Bloomer, for that presentation. So we'll start with a Q&A right now. And if you do have any questions, please put those in the question box. We'll start first with a question for Scott regarding plant proteins. So with respect to the growing industry demand for plant proteins and ingredients, what areas do you foresee being prioritized in industry?
Thank you, Susan. The big giant in the field, soy, has been around for a long time, and so there are very sophisticated methods that have been around, and the expectation of everybody else is that, especially on the side of the consumer, is that, well, you can measure this in soy, so you should be able to measure it in this new plant protein ingredient. However, they don't always act the same. So there are basically 5 big boxes that people are looking for methods in. And the first of all is the composition. Strangely enough, you can't just use a soy method for every other plant protein material. And I should say that, you know, people are looking at the composition, not just of the actual extract that you would get, but then concentrates and isolates, etc. Speaking about concentrates and isolates, there are specific definitions for soy proteins and concentrates and isolates in the Codex Alimentarius book, and they've got these official agreed upon standards, but the same does not apply in many of the other plant proteins. So there's kind of an anarchy in that people are allowed to call a product a concentrate because there's no standard for what a concentrate is, and they've taken something and concentrated it. So there's a real strong need just for simply labeling for composites, the composition, the proximate analyses. The second area of 5 areas that I see would be a sort of a structural, textural, aggregation, denaturation state. So this is kind of a part of the functionality in the mouth and in the actual processing. And so that would be a very important thing. So like elasticity, extrudability, and things like that. So then that's where you get into indicators of protein denaturation, protein fractions, particle size, a huge array of things. So that would be the functionality aspect. The third aspect is allergenicity. It's a very difficult sort of thing because it's a very big question and there are so many different possibilities. And you know, the truth is a lot of these proteins are not, they haven't been used by a broad number of people. A lot of them are very specialized and were common in one diet or another, but you expose that to a different genotype of human and there can be a variety of allergic reactions that nobody was anticipating. Of course, sensory would be the last. We touched on that already, but if it doesn't taste good, people are not going to eat a lot of it. It's just going to fall away and they'll reach for their chocolate protein bar that they've been buying for 10 years anyway. And then finally, we have the last category would be nutritional value. This would include anti-nutritional factors. And this again is sort of a big question and requires a lot of work. So we have got our work cut out for us. So I would say those 5 areas: composition, functionality, allergenicity, sensory attributes, and nutritional value.
Thank you so much. It sounds like, yeah, in the area of plant proteins, we definitely all have a lot of work to do you know, get these methods that are applicable for all parts of industry, as well as really encompassing the wide variety of proteins that are out there now, and not just the soy protein that we've traditionally used. All right, thank you for that. We have another question here. Have you ever considered developing the analytical methods using NMR? For example, using proton NMR for analysis of the MCPD in fatty acid glycidyl ester.
AOCS method development is divided into 5 different subcommittees. And we have a subcommittee devoted to rapid and non-destructive methods. And NMR methods fit right into that box. So the way that it works with method development organizations like AOCS is that somebody in the industry sees the problem and develops a method and then brings it to us. And then we take it through the process of the reviewing of the method. We have experts that, these 5 expert panels or subcommittees, we call them, that scrutinize the method, and they're very, very hard on it. There's a lot of back and forth between the method developer and these subcommittee members. And once that's done, then the method is ready for a collaborative trial. So then we have a study director frequently that would be the person that proposed the method, and then AOCS supports the study director in helping to design the study and finding laboratories and finding samples. And the study design is reviewed by the head of our Uniform Methods Committee. And when that person says, yes, it's ready to go, then we can launch the collaborative trial. Samples are sent out, results come back, and then AOCS analyzes those statistically. Xin Wu, who will be speaking next, is in charge of that. And she's meticulous and very thorough. And then after that, the whole package goes back to the subcommittee of experts who review the results in the study and look very closely at it. And so then there's another review by the Uniform Methods Committee, which is the highest body of methods experts that we have. And they, again, are very, very careful and rather demanding. And so there will be more back and forth, and finally, a method issues. So I would say, yes, there's room for an NMR method for anything that the NMR can be applied to. And a rapid method for MCPD and glycosylation esters would be very desirable. These process contaminants that we call MCPD esters and glycosylation esters are not really amenable to that. So it's going to be a difficult challenge. I'm not saying it's impossible, but it's going to be difficult to develop a rapid method for them. However, it would be wonderful. I hope I answered your question.
Yes, thank you. Yeah, I think it's important for everyone to realize it's not really just AOCS that's doing these methods. It's really driven by the people who are the members of AOCS. And so that, you know, if there is a method that someone wants, all you have to do is start and, you know, submit your idea and get the work going on it, because these are definitely all driven by people in the industry, people in academics all around, all the different products, you know, that AOCS works with. So thank you. We do have another one here. For non-saponifiable, will AOCS have a method to quantify tocopherol and sterols? I think this might be another type of question. You know, I think it's something, you know, it might be a need that's out there. And again, if there's a need, I think that you have to look into it and see how you can develop that method.
Yeah, that's exactly it. We already have some methods for these materials, and we find these frequently in, of course, deodorizer distillate. And there is currently a collaborative trial going on for determining the composition of deodorizer distillate. And so I would say, if you haven't already checked the AOCS method list online, you will probably find methods already there. But if you have a method, bring it to me.
Yes, and also I'll also plug too some of these questions around methods. If people have not been onto the Inform Connect site, this is a great place where if you have maybe specific questions on methods and you want to get an idea from others in the industry who like to collaborate on these things, a lot of people do like to post these types of questions on that. And there are some people out there who take great pride in their answers. And I think that they give some really good detailed answers when they can to people. So that's also a really good community to plug into if you have some of these types of questions. Okay.
That's a good point, Susan. We have recruited people for some of our ISO subcommittees based on their frequency of input into Inform Connect, answering questions. So.
Yeah. Okay. I'm not seeing any more questions pop up right now. I'm sure if you do have any other questions, the technical staff at AOCS is more than happy to answer your questions around methods and methods development. So please contact them anytime if you do have any further questions. So thank you, Scott. I appreciate this. And we will move on to our next talk then.
Thank you, Dr. Seegers.
All right, so next we have Xin Wu. Shen Wu is a technical services manager at AOCS. She is responsible for data analysis and reporting, method development, and quality control in the technical team. She ensures the laboratory proficiency program is compliant with ISO 17043, and the certified reference material program is compliant with ISO 17034. Before joining AOCS, she was an associate scientist at Intertech. She holds a master's degree in analytical chemistry from the University of Arizona. Now we have Shen Wu. We'll talk about the statistical treatment of proficiency testing data and how to interpret laboratory proficiency reports. Thank you.
Hello, everyone. Welcome to the presentation. My name is Shen Wu. I work in the Technical Services Department of AOCS. This presentation is prepared to be informative to potential or existing participants in the AOCS Laboratory Proficiency Program. I'm going to dive into more details on how a report looks like in our Laboratory Proficiency Program. The Laboratory Proficiency Program is one of the services offered by the Technical Services Department of AOCS. The Technical Services Department offers the official methods and recommended practices of the AOCS, the proficiency testing programs, and the certified reference materials. While the Certified Labs program focuses on soybean meal testing, the Laboratory Proficiency Program has about 40 different series. The Approved Chemist program and the Quality Reference Materials are extensions from the Laboratory Proficiency Program. From a participant's perspective, the Laboratory Proficiency Program goes through 5 steps: register, receive samples, test samples, submit results, and review report. In this presentation, I'm going to focus on the last part, review report. This report is published every quarter. The report has 4 parts: test results summary, summary of z-scores, chart of z-scores, and the kernel density plot. You could review your results submitted in the test result summary. You could find out how well you did in the summary of z-scores. The chart of z-scores and the kernel density plot are provided for better visual comparison of the results across labs. The first part of the report is the test results summary. This is where all the submitted results are listed. Here is an example of the test results summary page. When reading a report, the first thing I recommend to do is to check the top left and the top right corners. At the top left corner, there is the report title. At the top right corner, there is the sample name. When you are participating in more than one series, or when there are more than one samples in a series, checking the top left and the top right corners can keep you from reading the wrong report. The next thing to do is to find your submitted results in the report. Go down the first column on the left to locate your analyst number, and then go across to find your submitted results. Each constituent is assigned 1 column. To sum it up, in this first part of the report, test results are listed. The second part of the report is the summary of z-scores. This is where the z-scores and other summary statistics are listed. As always, you could locate the sample name at the top right corner. Let's first look at the z-scores. To find your z-score, go down the first column to locate your analyst number, and then go across to find your z-score for each constituent. Say if you find a z-score of 1.23. What does that mean? When the absolute value of a z-score is less than 2, it is considered to be acceptable. When the absolute value of a z-score is between 2 and 3, it is considered to give a warning signal. When the absolute value of a z-score is greater than 3, it is considered to give an action signal. In the report, when the absolute values of z-scores are between 2 and 3, they are highlighted in yellow. When the absolute values of z-scores are greater than 3, they are highlighted in red. Why do z-scores have such implications? When the measurements are carried out correctly and generate normally distributed results, the z-scores are normally distributed with a mean of 0 and a standard deviation of 1. The absolute value of the z-score tells you how many standard deviations you are away from the mean. Only about 0.3% of z-scores are expected to fall outside the range of -3 to 3, and about 5% of z-scores are expected to fall outside the range of -2 to 2. This implication is consistent with the calculation of z-scores. The z-score is calculated by taking the difference of the test result and the assigned value, and dividing that difference by the standard deviation. In the report, the assigned value is the consensus value. The standard deviation is the target standard deviation. Both values are determined according to ISO 13528. How about the other values in the summary statistics? The ones circled in blue are all calculated from the ones circled in green. For example, the relative standard deviation is the target standard deviation divided by the consensus value. The lower limit of tolerance is the consensus value minus 2 standard deviations. To sum it up, the second part of the report contains the z-scores, which are useful in monitoring the performance. The third part of the report is the chart of z-scores. It is essentially presenting the z-scores given in the second part of the report in a chart, where the x-axis is the z-score, the y-axis is the analyst number. The chart of z-scores makes the comparison of z-scores across labs straightforward. To find your triangle in the chart, locate the analyst number on the left of the chart, and then go across to find your z-score for each constituent. In the chart, the greater the magnitude of z-scores, the longer the length of the triangles. And the triangles are coded in different colors for their different implications. Blue for acceptable, yellow for warning, and red for action. To sum it up, the third part of the report provides charts for z-scores. The fourth part of the report is the kernel density plot. It is describing the distribution of the dataset. In the plot, the x-axis is the test result. The y-axis is the probability density. The peak of the plot is where the values are concentrated. The kernel density plot provides a visual summary of the reported values, and it contains information from the first and the second part of the report. The green cross here is where the consensus value is. And there's at least one test result associated with each circle, at where the values are concentrated, especially at the peak of the plot. Because so many values land at one place in the plot, one circle could represent multiple values. The color band at the bottom of the plot is coded similarly as the triangle in the chart of z-scores. Blue for acceptable, yellow for warning, and red for action. To sum it up, the fourth part of the report provides graphs to describe the data distribution. Now we have viewed the 4 parts in a report. The results are listed in the test results summary. The performance measurements, the z-scores, are listed in the summary of z-scores. The cross-lab comparison of z-scores are made easier by the chart of z-scores. And the data distribution is presented by the kernel density plot. In addition to the report we just viewed, there is also the proficiency report. The report we have viewed is published each quarter, and the proficiency report is published each year. At the end of each program year, you will see the proficiency report available. The proficiency report has the performance summary, which determines if an analyst is qualified for the approved chemist status. The approved chemist program is an extension from the laboratory proficiency program. The proficiency report has 2 parts, the proficiency report and the ranking report. The first part of the report is the proficiency report. The proficiency report lists the combined score, the number of missing constituents, and the root mean square z-scores calculated for each constituent. The combined score is an assessment of the performance over the year. It is the weighted average of the root mean square z-scores of the constituents. If an analyst has a combined score of less than 1.4 and has no missing constituent, the analyst is qualified to apply for the approved chemist status. The second part of the report is the ranking report. It lists the analyst number in order of increasing combined scores, and only analysts with no missing constituents are listed. Therefore, if an analyst listed in the ranking report has a combined score of less than 1.4, the analyst is qualified to apply for the approved chemist status. We have viewed the report and the proficiency report. We have discussed what information is available in each part of the report. Thank you again for coming to the presentation.
Thank you so much. So now we will have some questions. If you do have any questions for Shin, please put those in the question box. All right, we'll start off with, for Shin, what action should be taken if the result gives an action signal?
The action to take would be to investigate the results thoroughly. For example, first look for clerical errors. Check if the results submitted online are correctly entered. Check if the results used are with the correct units. Second, check the sample. For example, check the sample if the samples analyzed are the correct samples and are they in good condition. Third, check for possible operation errors. Look at the steps that could easily introduce an error and see if they are carried out correctly. Review region logs. Are the correct regions used? Are the regions in good condition? Also review instrument records. Are the instrument in good condition and performing as expected? And so on. So the action to take is to have a thorough investigation of the results, which should help for later correction and prevention.
Thank you, Shen. So it sounds like if anybody ever has results that are outside of the expected norm, it really is a practice to go through really every part of your lab, from just the data entry part all the way through, taking a step-by-step look through your method to make sure that everything is really following what should be done in the official method.
Yes,
So,
yes,
thank you.
exactly.
The next question is around how do you participate in the AOCS LPP and approved chemist program? So how does that participation, how can you actually do that participation?
So you can go on to the AOCS website. I can actually pull out the website. So you can go onto the website and navigate to laboratory services, and then click on the LPP, Laboratory Proficiency Program. So we have a list of programs, around 40 programs. You need to go through that list to find what you need and follow the steps, and you can get registered and start participation. And for the approved chemist program, the same thing. Go to the AOCS website where you find the LPP. You can also see the approved chemist program. So you can, it's after participation in the LPP program, you get to be able to participate or apply for the approved chemist status.
That's right. Yes, so it's really taking the status and how well you've done in the LPP, and then using that then to apply to be in the to be an approved chemist. So, and again, like Shen said, the AOCS website has all that information, and anybody there would be happy to discuss with you on how to participate in that. And I know from experience, we've participated in that for many, many years, and it's a great program that really is helpful for the lab. And I'm also the chair of the LPP, so I'll put that plug in there too, because it is, like I said, it's a really, really good program, provides great results for your lab, to be able to look at data that's compiled from many people in industry and other labs, doing the exact same methods you are, to really make sure that you are in agreement with labs as a whole. Okay. That is all the questions that I'm seeing here right now. I will just say again, thank you to Shen for presenting this. I think the LPP is, like I said, it's very helpful for everyone. And the data that we get out of the LPP, I think, is so helpful, and just the great way even the reports are presented, I think, is a very actionable and thorough way that we can see what the data looks like. It's very visual. I really appreciate the reports that come out of that program too. Okay. If there are no other questions right now, then I think we will adjourn this session. I thank everybody for joining today, and I thank Scott and Shen for their presentations. This was very helpful, very informative, and I hope it sparks more conversation around AOCS methods and statistical treatment of this data. I think these two are very, very important areas that AOCS really is helpful for our industry in. So again, thank you all for participating, and thank you, Scott and Shen, for your presentations.
Thank
I
you,
wish everyone had a great day.
Thank you, Susan. Thank you.
Thank you.
Bye-bye.
Bye-bye.